All right, thanks everyone for joining us today for our symposium. I'm going to start by talking a little bit about reproducibility. So, most scientists in biomedicine feel like we're experiencing at least some issues with reproducibility, even if you think the term crisis is a bit much. And major agencies agree. Federal agencies like the NIH, companies like Amgen, and we've discussed this extensively in neuroimaging as well. So, what can we do about that? A major theme is that we can achieve better reproducibility with more accurate methods. Accuracy can be thought of as a combination of reliability and validity. Reliability refers to the ability to get similar results given repeated trials, and validity refers to your ability for a measure to correspond with some desired outcome. So let's say we have an archer and we want to see whether they're accurate, and we let them shoot. So we can see that the arrows clustered a lot, which means that it's quite reliable, but they're not quite on the bullseye, which is in this case our desired outcome, suggesting that it's not necessarily valid. If instead they shot like this, where the arrows actually on average would hit the bullseye, or at least the average of those positions hit the bullseye, but each of the arrows are really separate from one another, we would say that this is perhaps more of a valid shot, but not very reliable. And especially in this day and age, I think a good example is testing. I think testing helps illustrate how both reliability and validity are really important simultaneously. So if you have a test, like in the first case, that isn't accurate or that isn't valid, that doesn't tell you the actual outcome, it doesn't matter how reliable that test is. It's not going to be an accurate test, or it's not going to be a test that you want. If, on the other hand, you have a test that's valid on average, that on average gives you the correct result, but each time you go in to take the test, it gives you a different result, and you need to do it 100 times and average that together, then that's not very reliable, and that's not that useful either. So both of these are very important, but today I'm going to focus on reliability, and specifically reliability of functional connectivity. Over the past decade, many investigations have taken place investigating this very issue. So starting with Shahzad and colleagues in 2009, and then these are just a very small sampling of the very vast literature on this topic. However, results are mixed across all these studies, and they're further complicated by the fact that people are studying very different things, which is great. But I think that there are three main questions that we want to address. How reliable is functional connectivity? And we've had reports ranging from poor to excellent. How do we improve functional connectivity? Some studies say that we should do this strategy, and other studies say that that strategy actually makes reliability worse. And how useful is this? Should we purposefully be going about studies that improve reliability? And I think these are all still open questions. And we need to summarize this rich literature to better answer each of these questions. So the following, I'm going to show you a review and meta-analysis and a few previous studies that discuss these questions. So I'll start by telling you how I went through and searched through the literature. And here's a Prisma diagram. So our inclusion criteria, where we were looking for fMRI-based connectivity at this individual edge level in healthy populations that looked at the whole brain. And just to be able to compare one study to the next, we measured reliability via the ICC. So after the initial search terms, we had 213 studies, and then further reviewing the full text, we ended up with 44 studies for the qualitative synthesis. And for the meta-analysis, we included those studies that reported the mean and standard deviation of the ICC. And we actually reached out to other studies from the qualitative synthesis if they did not report that, to try and get as many numbers as possible. But 25 studies in the meta-analysis. And since we're talking about the ICC, I'm going to go into this in a little bit more detail. So the interclass correlation coefficient, or the ICC, measures relative variance due to person relative to error sources. So it's actually due to the object of measurement, but typically in neuroimaging, that's person relative to error sources. And here I'm showing you the very simplest form of an ICC, which is the ICC 1-1. There are many different forms which we discuss in the review and meta-analysis, so please check that out if you have more questions. But generally speaking, if you increase variability due to person, which means you make people look really different from one another, And you decrease variability due to error sources, which means every time you measure an individual, they look a lot like themselves. Reliability will approach 1. If the opposite is true, where we decrease variability due to people, which means each time different people are starting to look like one another, and we increase variability due to error sources, which means every time you measure an individual, they start to look like a different person. Then the ICC will approach 0. So 0 to 1, 0 being the worst, 1 being the best, and we have this kind of rule of thumb for interpreting different levels of ICC, where the lowest you can get is below zero point 0.4. So now that we understand the ICC, let's look at what studies over the past decade suggest about reliability, about the ICC of edge level connectivity. So here's our meta-analysis, and we made a few assumptions here about the distributional nature of the ICCs. There actually are, we weren't able to find any other studies that did a meta-analysis of ICCs. Some studies use ICCs in the meta-analysis, but don't do the analysis of the ICCs themselves. So we made a few assumptions here. Please check out the paper for those details, which I can't go into right now. Um, and we came out with a point estimate of 0.29 with a confidence interval from 0.23 to 0.36, which suggests poor reliability at the individual edge level. Just because we made some of those assumptions, just for comparison, we also did an arithmetic estimate, a very simple pooled estimate, and came up with an ICC of 0.38 plus or minus 0.09, which brings it slightly more to the boundary between poor and fair. Um, so you can just check that out if you're interested in the differences. But overall, seems to be more so on the poor side. And even if overall reliability is poor, perhaps some areas are more reliable than others. So this is just a ranking by consensus. So most studies seem to suggest that the frontal parietal and default mode networks were most reliable, followed by these kind of sensory areas, visual networks, sensory motor, and then more subcortical areas, limbic, cerebellar, and then areas that are adjacent to non-gray matter sources like CSF. And then more general subcortical areas were ranked least. So this kind of somewhat to be expected axis from these large cortical networks to subcortical areas. Now, what are the effects of various acquisition and processing strategies on reliability? Multiple sites and scanners seem to have a minimal effect if they are harmonized. Doing eyes open, awake, and engaged recordings seem to improve reliability slightly. Slice timing correction seems to have a minimal effect. Having more data per subject, so that's both, longer scans, higher TRs, or sorry, lower TRs, higher temporal resolution. Those can improve reliability, but they have diminishing returns. So the more data that you have, you'll start to have fewer and fewer improvements in reliability. Having a short interscan interval improves reliability. And then this one I think is very interesting. Noise regression has very mixed effects on reliability. So people have investigated many different types of noise regression, global signal regression, different types of component regression. Um, the majority of the literature seems to suggest that reliability decreases with these various techniques, but it is mixed. And I think it's very interesting because it's complicated by a couple factors. First, artifact itself, like motion, has been shown to be reliable. And also, denoising has been shown to improve meaningful outcomes. So, more on the axis of validity rather than reliability. Even when I think the Shearer and colleagues studies is very interesting, they show that reliability can decrease even when these meaningful outcomes increase simultaneously. So, kind of interesting, noise regression can decrease reliability or have mixed results, but simultaneously improve validity. Using partial correlation has slightly lower reliability than full correlation, and connectivity improves reliability when estimated with shrinkage. It's kind of interesting span of results from various acquisition and processing strategies. Now I'm going to talk about how a few of those relate to some findings from a previous study. You can check out that study below, and we have some data down there at the bottom that you can check out. So first, you can do what's called a decision study to estimate how reliability changes with different amounts of data. Here I have scan duration versus number of sessions. And you can just see in this figure, so blue is least reliable, bright colors are most reliable. And you can see that there's some asymmetry here, suggesting that 36 minutes of data at a single session is much less reliable than 36 minutes of data across 6 sessions. So 6 minutes per session. That's just one thing you can do with a decision study. And then back to this question of reliability versus validity or utility. Here I plotted the behavioral relevance of edges in predicting fluid intelligence against their test-retest reliability. And you can see already these spatial patterns are quite different. The correlation between these two measures is quite low, and actually reliability explains less than 1% of behavioral relevance. So just adding nuance to that question. In conclusion, what have we learned about this past decade of investigation to the reliability of functional connectivity? So first, we got a more accurate point estimate of test-for-test reliability at the edge level, and it still is quite poor, as some studies suggest. However, we can get better reliability um, by looking at stronger within network cortical edges, eyes open engaged recordings, more within subject data, shorter test retest intervals, no artifact correction, but again, that comes with caveats, and full correlation based connectivity estimated with shrinkage. But it's very important to remain careful. Reliability is not utility. There can be a trade-off, like we saw in the artifact correction section. There are still open questions there, but it's important. It is important to improve both. We do want both reliability and validity, but we don't want reliability at the expense of validity. So just keep that in mind. All right, so I'd like to thank everybody who helped out with this project. Dustin Chinos in the Mines Lab, Todd Constable in the Constable Lab, Ty in the Naples Consortium, HCP, and my funding sources, and my current team, which is the BISWeb team. Thank you. Any questions?
So, uh, so, so in this recording or in this symposium, we've been speaking a lot about measuring the individual or looking at individual differences or sources of reliability or variability across individuals. And one reason why this really matters to me, especially as someone who does clinical or neuropsychiatry research, is that in our research, we focus a lot on trying to understand how to actually minimize some types of variability so that we can actually measure other types of variability across people. And the two processes that we use to do this are two processes known as realignment and sometimes parcellation. So if we think about realignment, if we think about the derivatives that are available, especially from the Human Connectome project, we actually have quite a few choices to look at. So the first type of derivative is one that we're very familiar with, and that is data that's been realigned in the three-dimensional volume into M&I coordinates. But in the Human Connectome project derivatives, we also have these files that are in zip format where data has been mapped to the surface and realigned on the cortical surface. And then when we actually think about cortical surface realignment, we have additional options. So there's some outputs where data is realigned based on sulcal depth or anatomical data, but then there's additional output where data is realigned based on additional features, including additional anatomical features, task-based fMRI, and resting state fMRI. And these derivatives, or these files, because they've been realigned on so many things, we refer to them as the MSM-all, or the multimodal surface alignment all derivatives. And then when we pick which type of derivative we want or which type of realignment we want to use, then we usually go forward and do a second process of parcellation. And we have a lot of parcellations to pick from. Some have been developed in the volume and published in the volume, and some have been developed on the surface and have been published on the surface. And then some of those developed on the surface have been, again, mapped into the volume. But one thing that I wanted to make clear is that every time that we actually move from the volume to the surface or move a parcellation from the surface to the volume, we're actually lying to ourselves a little bit if we think we're doing an amazing job. The problem is that everybody's brain or everybody's brain has a slightly different folding pattern. So when we try to map from the volume to the surface across multiple individuals, what actually ends up happening is we get a different result for every person. And because we get a different result for every person, when we go to try to get a population-based result or a sample-based result, what we actually get is not actually a perfect result, but a probabilistic map on the surface. And then if we take one atlas or multiple ROIs within an atlas or multiple parcels within a parcellation, and we try to get a group result, what we end up getting is, um, for example, by taking the maximum value across multiple parcels, we get a map that looks like the one that's on the right. When we look at this map, it has some really interesting properties that we should actually really care about as neuroscientists. So one interesting thing is that there's some areas of darkness here, or some areas of the brain that are actually much harder to map from volume to surface. And that's actually because those areas of the brain actually show the most variability in sulcal depth across the population, or the most differences in people across the ways that our brains fold. What's important about these areas is they actually also have different cognitive properties. So if we think about the brain in terms of a network approach, this is the brain actually parcellated according to the Schaefer 400 parcellation. We see that some of the networks, especially the networks for visual attention and the networks for cognitive control, actually are the most difficult to map from the volume to the surface, and then again from the surface to the multimodal version. If we care about the brain, or if we want to think about the brain not in terms of networks, but instead in terms of gradients, actually following from the work of Margilis in 2016, we actually noticed that this difficulty mapping from volume to surface also follows cortical gradients, especially the fourth gradient. And if you want to hear more about these properties, you can visit my poster number 1827. So we might say, we saw that this is different across the surface, but we might ask, does this really matter? Will this affect the way that we actually see brain behavior relationships or differences across people? So to investigate this, I actually took data from 425 unrelated individuals from the HSP dataset, and I took those three different types of derivatives, so the volume-based derivatives, the anatomically aligned derivatives like the MSM-SULC, and the multimodal realigned, the MSM-ALL, and I parcellated them according to every parcellation I could find, including all of the sizes of the Schaefer atlas, which is actually available. You can apply to all three of these different types of derivatives. And we used linear ridge regression in scikit-learn to try to predict certain variables. In this case, this is age group and gender. And across multiple variables, we noticed this pattern. This is for 4 different cognitive scales. Applying the same process, we noticed that the volume-based derivatives plotted in red seem to be usually the poorest performing, which means that surface-based derivatives may be very useful when it comes to actually thinking about brain behavior relationships. And again, you can visit my poster for more information about this study. So moving on from that work, which was in healthy adults and thinking about cognition, I want to actually take this more to a clinical research side. So one study that we've been working on includes data, investigated the effects of these mapping techniques on research, looking at schizophrenia case control differences using resting state data. So to do this research, we've been using 4 different cohorts. We have a cohort from my colleagues at the Center for Addiction and Mental Health, and another cohort from colleagues of mine at Zucker Hillside Hospital in New York. We also have 2 publicly available data sets, the COBRA study and the UCLA Consortium for Psychiatric Genomics. From all 4 of these cohorts, we have data from schizophrenia cases and controls, and I pseudo-matched across cohorts for age and sex to using the Match-It program in R. So we have a total of 200 cases with schizophrenia and 206 healthy control, pseudo-matched healthy controls. And from all participants, we have less than 7 minutes, in most cases less than 5 minutes of resting state data at the kind of parameters that we've been using for the last decade. So from this data, I wanted to see whether or not mapping methods would actually affect the kind of results that we saw. So I pre-processed the data using a pipeline where I'm actually the lead developer. It's a zip file pipeline. It's available as a BIDS app publicly. And you can read about it more in my neuroimage paper. And it essentially mimics many of the pre-processing steps that are applied in the HCP pipeline, but will work on hopefully any data set. So from this data, we have the volume-based or the MNI-based derivatives, and from those I extracted data from 80 MNI coordinates or 80 6 mm spheres surrounding MNI coordinates, and the location of these spheres were picked specifically to try to measure from 6 resting state networks of interest. And then on the surface, or the anatomically mapped surface data, I picked 80 similar points, actually the same points, but in this case, we're measuring from circles on the surface that are 6 mm in diameter. And then to look at a more multimodal realignment technique, I used a technique for individualized parcellations called personalized intrinsic network topography, which is a method that I've published on in the past, which is also available with the Siftify package publicly. And it actually takes those points on the surface and moves them on individual surfaces to points that will maximize within network correlations. To show you what this means, I'm actually going to show you some data from some individual representative subjects. So these correlation matrices plotted here are actually extracted from the volume, the surface template, and the surface personalized data from individual representative subjects in our data set. And here is the mean correlation matrices from these 80 cortical surface ROIs. And you can see that in the volume template, in the volume version, we do see some evidence of resting state networks, so some evidence of darker, redder squares along the diagonal. But this is very, very much increased when we go to the personalized pint mapping technique. That's not that surprising because that's what pint is meant to do. But the real question we want to think about is if we send these matrices, if we use these matrices to investigate schizophrenia case versus control differences, do we get a different result from if we use these matrices? So to investigate that, I did edgewise I calculated edgewise case control differences, working from this data, and I have it plotted in a little bit of an unusual way. So I calculated the edgewise case control differences in linear models that included age, sex, in scanner motion, as well as scanner as covariates. And then results here are presented, results or edges presented in this plot are those that survive FDR correction. And the red edges are the ones where I saw evidence of hypoconnectivity or a stronger correlation in schizophrenia participants relative to healthy controls. And the blue edges are those where I saw less correlation or less connectivity in the schizophrenia, in the participants with schizophrenia relative to health controls. I have these edges organized in this way, which I like, and I'll explain why. So all of the cortical ROIs are along this line in the middle, and they're organized according to cortical hierarchies. So we have default mode, followed by control or ventroparietal network, followed by salience network, followed by dorsal attention network, followed by somatomotor network, followed by the visual network. And then at the bottom here, I have some subcortical regions that are parcellated according to some published parcellations of the cerebellum, the thalamus, and striatum. I like organizing my results this way because it means that all of the cortical connections are plotted up in the top right. All of the subcortical connections are plotted in the bottom right here, and all of the subcortical connections are plotted on the left side. So we can already see that some edges, using this is the volume sphere-based result, are significantly surviving FDR correction, and that they have some patterns going on. But let's look at what happens when we switch to the surface-based anatomical result. And again, let's see what happens when we switch to the personalized result. So what I really like about the surface-based and the personalized result is that it seems that we're good. Sorry. It seems that we're actually getting cleaner and more interpretable results in this case. So we see this huge swath, and it got darker, of red in the bottom here, and this is expected patterns of hyperconnectivity in schizophrenia that we see between the thalamus and the visual and somatomotor cortices. We also see this huge band of blue edges, and this is expected patterns of hypoconnectivity between the frontal parietal network and the subcortical regions. So to summarize, the reasons why I really like working with surface-based data and multimodal-based data is first because I want to make it clear that volume and the surface are not actually the same thing. So when we extract from the surface, we especially potentially get a better measurement from areas of visual attention and areas of executive control. I hope you also saw that in both the HCP sample of healthy adults and in our schizophrenia case control differences, we saw stronger predictions or stronger predictive power. We also saw more interpretable results when we used the surface and the multimodal perspirations. So I think now there's actually a really strong case that methods really do matter, and that we should really move towards these surface and these multimodal techniques. The real question is how, and I think we actually need much stronger education, and we actually need to do a lot of work to try to make these techniques as easy to use as volume-based techniques. There's been some work going forward. There's been a lot of work, including my work with the Siptify package, to make it easier to pre-process data and get surface map derivatives. And there's been a lot of work on individualized mapping techniques that have been published and made publicly available. But I think there's actually a lot of work to go because there's so many techniques and some of them actually don't work together very well. And there's a long way to go in terms of education. But with that, I'm really excited to be working with my lab and working with everyone who's been adopting these techniques with me. And thank you so much for listening to this talk.
All right, greetings from Evanston, Illinois. It's a real pleasure to be part of this symposium looking at measuring the individual, where I bet you've been hearing an impressive series of talks prepared by my colleagues on measurement reliability, cross-subject anatomical alignment, variability in task activations, and other important considerations in how we might study individual brains. And I feel fortunate to have the opportunity to tell you a little bit about our work looking at variability in human functional brain networks using a precision approach. So, as I'm sure you've heard throughout this conference, fMRI functional connectivity presents us with this really amazing ability to provide these beautiful, detailed pictures of functional neuroanatomy in the living human brain. Just to be clear about the methods I'm using here, when I talk about functional connectivity, we are just meaning taking correlations in the bold activity signal between different brain regions. For example, here I'm showing you the pattern of bold time series between left motor cortex and left visual cortex. And you can do this for many different regions spread throughout the brain. And by looking at the regions that share common signal, identify networks or systems of regions throughout the brain, which I've painted in different colors as shown here. Moreover, we've recently come to understand that not only can we make these measurements at the level of the group, as I've shown you on the previous slides, but with sufficient data, you can also make these measurements quite reliably at the level of individual subjects, as is shown here for 8 of the subjects from the Midnight Scan Club dataset. When you do this, what you end up finding is that each of the individuals shares a lot of features with the group, but also has unique features that are specific to their own brain organization. So this observation begs the question of what do these individual differences in functional brain networks indicate? That is, what are their characteristics? In a series of projects I'd like to tell you a little bit about today, we've started probing deeper into the characteristics of these individual differences. The first project was spearheaded by Ben Seitzman and asked the question, are individual differences in functional brain networks trait-like markers of underlying differences in brain organization? If so, we hypothesize that they might have certain characteristics. First off, that they should be stable over time, so a single measurement within a given individual looks most like themselves at other moments in time. Secondly, that they should present systematically across people with separate subgroups of individuals within the population that have similar phenotypes. Third, that these individual differences in functional brain organization would also relate to individual differences in function. So we used a combination of a deep data set and a big data set in order to address this question. The deep data set was the Midnight Scan Club data set I introduced earlier, which contains data from 10 highly sampled individuals who each came in for 10 separate fMRI sessions, accumulating approximately 5 hours of resting state data and 6 hours of task fMRI data. The second data set is the Human Connectome Project data set, from which we used data from 384 high quality individuals who had more than 45 minutes of good resting state data, as well as a number of neuropsychological measures that were collected outside of the scanner. Ben devised a very straightforward way to then identify the locations that were most different between an individual and the group, locations that we call network variants. To do this, he went point by point through the cortex and created a seed map for each point, as indicated for this point at the white circle at the tip of the arrow here for subject MSCO6, and compared that individual seed map to the typical group seed map at that location. When you look at these, you can note that they're quite similar to one another. So when you take the spatial correlation between the two, as you'd expect, it's quite high and colored by a warm color in this map. Another subject, however, here MSCO2, might have a very different pattern of correlations for this point, known in particular the anti-correlations with lateral frontal cortex. And so the spatial correlation ends up being quite low and colored by a blue color on the spatial correlation map. So we can make these measurements for many regions spread throughout cortex, all the regions spread throughout cortex, and then threshold this map in order to identify those locations that are most different between an individual and the group. And that's what we call network variance, as shown in this map at the bottom. You'll note that I've also colored the network variants in different colors depending on which network they now associate with. So now we turn to our original hypotheses about whether or not these network variants would be trait-like markers of different phenotypes in brain organization. So first, we looked at stability over time. What we find in the Midnight Scan Club is that these network variants are quite stable over time. They appear again and again across the 10 different sessions that the participants completed, as noted by the same blue locations, especially the circled ones, appearing across this subject MSCO2. And also as indicated by the high interclass correlation coefficient for these maps in the subjects from the Midnight Scan Club. Note that one subject, MSCO8, had much lower interclass correlations. This subject had much less data from each session due to high movement and sleep issues. I'm happy to talk at the end more about how much data we think we need in order to make these measurements reliably. And this subject is excluded from future analyses. Moreover, we've recently had the opportunity to expand our look at stability beyond the few weeks that was collected in the Midnight Scan Club to look at also stability over the course of the year in the My Connectome project. What we end up finding is that these network variants are also quite stable over this time window. As you can tell, the network variants are very similar between the first 3 weeks and the last 3 weeks of that year. My graduate student, Brian Krause, has also recently looked at whether or not network variants can be identified across different states, and in particular across different task states. And again, we find that although there are differences between rest and task states, that the locations of network variants are quite similar between the two, as indicated again by the similarities in these spatial correlation maps and the overlap of the final network variant maps. So in the next set of questions, we looked at whether or not these network variants present systematically across individuals. First, we asked where they tend to occur in the brain, and this is shown for the Midnight Scan Club subjects, the overlap plot of the network variants across those 9 individuals in that data set. What you find is that they're not spread evenly across the brain, but rather tend to appear in certain locations like the temporal parietal junction, as well as lateral frontal cortex more frequently. Quite nicely, this pattern also replicates in the Human Connectome Project dataset, and this is a notable replication because these are not only different individuals, this was also collected on a different scanner with very different spatial and temporal resolutions. Next, we asked whether different people might show shared phenotypes of brain organization. That is, do they show similar network variants to one another? And we find that indeed they do. Interestingly, their similarities are not driven based on where the network variants appear, but rather which networks these variants are part of. So what we end up finding is that one subgroup of individuals, shown here in gray, has network variants that associate more strongly with sensory motor processing and control networks, as shown here. And another subgroup, shown here in pink, has network variants that associate more strongly with a default mode subgroup, as shown here. Again, these two subgroups came from the Midnight Scan Club, but we replicated this pattern in a larger sample from the Human Connectome project, as shown here. In this larger sample, it also becomes clear that there's likely additional, or there's hints that there's likely additional subgrouping within this sample. And indeed, we don't mean to suggest that there are only two phenotypes or two subgroups of individuals based on their brain organization, but rather that this is a proof of principle that these network variants can be used in order to subgroup individuals into different types. We also were interested in looking at whether or not these individual differences in brain organization were related to individual differences in function. We approached this in various ways. I'll just tell you about one today, which is to look at whether or not these individuals in the different subgroups I just introduced also differed in their behavioral measures collected outside of the scanner. And indeed, what we find is that those in the default mode subgroup showed lower positive life experience and higher history of drug abuse than those in the sensory motor processing and control subgroup. So just to summarize, what I've shown you here is that network variants are quite stable over time, even up to a year, and that they also show some stability across task states. That network variants are more frequently found in association systems of the brain and can be used to identify subgroups of individuals with similar patterns of brain organization. And these subgroups of individuals also differ in their behavior collected outside of the scanner. So these qualities, their stability, their systematicity, their relationship to function, suggest that network variants may be trait-like measures of brain organization. In the last few minutes of this talk, I just wanted to also present a teaser on some of our more recent work, looking at whether or not there are different forms of network variants. This work has been led by Ali Doretsky. Ali's been looking at whether or not network variants that differ based on whether they're border shifts. So you can see here some network variants occur right on the border of networks, this yellow territory taking over parts of the purple territory. Whereas other network variants seem to occur at islands of locations far away from their normal network assignment, so which we call ectopic intrusions. So you can see that this red location occurs far from normal red default mode locations. And so Ali's asked the question, do these two different types of network variants differ in their characteristics? And again, just to hint at the results, what she's ended up finding is that both types of network variants are relatively common. They're both present in most subjects. However, that they do differ systematically in their anatomical distribution, with border shift variants occurring more along the temporal parietal junction and ectopic variants occurring more in lateral frontal cortex. And also that they seem to differ in their subgrouping, such that one person could be in a default mode subgroup for the border shift variants, but in a control subgroup for the ectopic variants. So these findings together suggest that different forms of network variants may be indicating different phenotypes for functional neuroanatomical differences. So I would like to close by thanking my collaborators on these projects, my former mentor Steve Peterson and Ben Seitzman, who have carried on a lot of the initial network variant work, as well as my current lab members who are carrying forth some of our more recent projects. And I'll again put up my contact information on the screen in case you want to get in touch outside of the course of this session.
Hi everybody, and thanks for joining us today for our symposium. My name is Dr. Colin Hawco. Today I'm going to talk about clustering task fMRI in healthy populations in schizophrenia to look at individual variability. This work arose from an emerging idea in psychiatry which we call biotyping, which is the idea of finding biological subgroups across disorders. This idea came out of the fact that our diagnostic labels in schizophrenia and psychiatry in general tend to be quite unclear and heterogeneous. So patient groups are very variable. The diagnostic labels haven't proven to be particularly useful scientifically. And so we're shifting our perspective now to move towards a more biologically derived way to identify groups of patients that share similar underlying biology, under the hopes that they'll have then similar profiles of response to different treatment interventions. But even in the field of general cognitive neuroscience, we can see that while we can calculate relatively reliable and static group maps, individual variability around these group maps can be quite high. So what does a group map really mean if a participant may or may not actually match that pattern of activation we see at all? And in the field of functional connectivity, while we can produce relatively robust and stable group aggregate averages connectivity patterns, individuals can look extremely variable relative to that sort of group average. So the group might not capture all the variability that's really of interest to us as we begin to explore human brain function. So the first study I want to talk about today is we're using hierarchical clustering using an imitate observe task in the fMRI. This work part of a large multi-site study that we're collecting data on. It includes patients with schizophrenia spectral disorder and healthy controls. We performed a hierarchical clustering approach using a task fMRI examining lower-level social cognitive processing, in this case, the facial emotional imitate observe task. In this task, participants are shown a series of emotional faces, a neutral face or a fixation cross. And in one block, they're asked to imitate the face of the scanner. We literally have them going inside the MRI. And in the other block, they're asked to just observe the faces and process the emotions. And this task has some nice features, in particular that it activates very reliably and robustly across studies as canonical so-called stimulation or mirror neuron network in the brain involving frontal regions. Also, we find this task shows reduced activity in autism with social processing deficits. Those task reductions, those activity reductions are related to their social cognitive deficits. And so when examining social processing in schizophrenia, you also show some deficits of this kind. This was a very good task for us to tap into the system. So what we did in this study is we took a subsample from an earlier phase of that study, looking at 109 schizophrenic inspections and 70 controls. And we calculated individual maps for each participant. We then express the spatial activity of each of those maps as a vector, and we stack those vectors by participant, and we can perform clustering on them. And when we do the clustering, this is what we get. At the top of this, we see the dendrogram, which is the cluster tree. We found a 3-cluster solution best fit the data. A couple of important points: there was no differences by cluster across sites, and very critically, from my perspective, no difference in cluster by diagnostic category. The people with schizophrenia didn't form their own little subgroup over here because we're all a lot more likely we are different, despite the fact that some people have psychiatric diagnoses. And if we zoom in and look at the detail of these clusters, we find this central group as what we call our typical activators. They show the pattern that we sort of expect to see in this kind of task based on those studies. But here on the right, we have this group that we think of as hyperactivators. They're showing an overactivation of the brain. We think this is sort of an inefficient pattern of activity. And here on the left, we have a group of deactivators, all this stuff in blue. And these participants shut down most of their other brain systems, and they minimally activate this sort of core canonical stimulation network. This is a pattern we see with expertise. For example, if you show a ballerina, a movie of people doing ballet moves, they'll tend to look like the deactivators. And if you show me that same movies, they'll tend to look like a hyperactivator because I'm not very good at understanding ballet. When we think about this behaviorally, we did find, in fact, matching our idea that this deactivation pattern was an efficient brain pattern, those participants tended to have better out-of-scanner social cognitive abilities. And while the patients in that deactivating group were still impaired, they were the least impaired of our patients. So this kind of fits the idea that this is a kind of an efficient pattern of brain activation who are good at this kind of task. Now, it's a great result, and we're very happy seeing this sort of spectrum from sort of low to middle to high. But our first question, of course, is how reliable and replicable is this? Happily, we had a replication sample available, in this case from a single site, looking at controlled schizophrenia spectrum or bipolar disorder. And when we did the same pipeline with the same task, we got, voila, very similar results. A deactivating subgroup, a typical activating subgroup, and a hyperactivating subgroup. So that suggests that this finding is quite robust and reliable over time. Now, this was very exciting for us, these kind of results. Obviously, biotyping is a very big deal in psychiatry. But if we're going to move into clustering as a major research approach, I wanted to really dive deeper into how stable it is, how reliable it is, and what we can learn from it. So in order to really dig into the methodology a little bit more, we used the Human Connectome data set, both from the HCP 900 release, which is 900 participants. And HCP includes 6 task domains, which gave us the opportunity to look at clustering across several cognitive tasks. And what we did is we took the 8mm smoothed HCP provided T-maps. They did all the first level statistics for us, which is quite nice. And then we're clustering each task separately, again, stacking your participants into a matrix using hierarchical clustering of wards method, which is what we find works really well in high-dimensional fMRI data. And we calculated and examined all solutions from 2 clusters to 10 clusters. For purposes of time and illustration, we're going to focus on K equals 4, the 4-cluster solutions mainly, because those were the most sort of informative. And we're also going to focus on the social task and working memory task, just so we can get through this in 10 minutes. This work is available on bioRxiv. We uploaded the preprint, so I will refer you to the preprint if you're looking for more information. If you go to bioRxiv and search my last name, Hawco, it should be the top hit. So when we look at the clustering results across this task, we see something very similar to what we found in the imitate observe task in the psychiatric patients, which is a pattern from deactivator, intermediate, up to sort of a stronger activating or more activating group. This was true for all 6 tasks. We can see quite, I think, clearly here in these 2 tasks. On the left side of the screen, over here, we have the total sample group map. So if you average everybody together, one important point I think is very salient here is that the deactivating clusters and the most strongly activating clusters don't necessarily match these group maps very well. So if we just look at the group aggregate averages, we're actually missing quite a bit of information in various data. But it does look like this pattern from sort of most negative to most positive is sort of prominent across a variety of tests we can measure inside the MRI. But our real goal here was to think about questions like, what information can we really pull out of this clustering? How stable and reliable is the clustering? How can we really use it moving forward? So to interrogate this further, we used a bootstrapping approach. We ran 1,000 permutations of bootstraps, rerunning the clustering on a randomly selected 75% of data with no replacement. Then we compared participants across bootstraps, and this produces a matrix something like this. This is a participant cluster agreement matrix or a bootstraps probability matrix. It's a participant by participant matrix. It's similar to a distance matrix or a similarity matrix. But what it measures is if 2 participants were present in a given bootstrap, what's the probability that they cluster together? Now, if clustering was perfect, we would see a series of white squares here, which means that the same people always cluster together in the same clusters. Instead, what we get is something like this. Across our 2 tasks and our different values of k going across the bottom of 7, we see something that looks more like pseudo-clusters or pseudo-spectrums. There's kind of boxes, but the boxes aren't really clear and distinct. And the color yellow there suggests that most people aren't clustering with more than about a 70% probability all the time. So across iterations where we're in the clustering, people can jump around quite a bit between clusters, but usually kind of to adjacent clusters and not fully across between distant clusters. So that's very interesting, and it suggests that there's information here, but it might not be clear to say you're in cluster 1, you're in cluster 2. So to really dig deeper into this, we took a principal components analysis approach. So we ran a PCA of this matrix, and this gives us components. So here, for example, this component seems to represent kind of the diagonal. This component 2 seems to represent kind of the bulgy outside position. And now that every person has 2 component scores, we can plot all of our participants on a scatter plot. So each circle here now represents 1 participant. The colors are the original 4 cluster solutions. So we see there's a kind of a cohesion here from what we originally saw. And if we add a 3rd component, we're now explaining 90% of the variance in our cluster probability graph. But something kind of cool happens is this circle kind of folds. It forms this sort of folded circle. I think of it as a taco pattern or a tortilla shape. And I think this represents now for us a lower dimensional similarity manifold space. The bootstrap approach is actually a data-driven feature selection stage. People are grouping together based upon the salient features of their brain image that is defined by the clustering algorithm, which just brings people far apart or separated. Now we can see how close different people are across different tasks. So if we zoom in on this a little bit, we look at this pattern of variation in this new sort of similarity manifold space, we can see, in this case, what I did is I somewhat arbitrarily chopped people in different sections of this graph up into subchunks, saying, well, if I look at this corner, what do people in this corner versus this corner look like? And the group maps came out like this. The horizontal axis here is our positive to negative axis. The people on the left are the deactivated, the people on the right are the activators. But the up and down axis, the vertical axis, shows something kind of interesting, which is the people on the top tend to have a combination of task positive activation in yellow and task negative activation in blue, weighted maybe one or the other, but still both present. The people on the bottom tend to have either task positive or task negative activation. So this second axis represents whether people are making use of either or both. And it happens that the second axis, this vertical axis, actually correlates fairly well with cognitive abilities outside of the scanner. We also found that 2 characteristic patterns emerged from the data. We either saw taco-shaped folded circles or snake patterns. I don't have time to go into the details. Again, I refer you to the preprint, but it looks like the snake S-shaped patterns are driven by sort of mean brain activation, sort of one system that goes up or down, versus the folded circle taco patterns are related more to if there's 2 or 3 independent brain systems that can each fluctuate up or down on their own. So they can both be high, one low, or vice versa across different individuals. And that gives rise to the circle pattern. Now we go back to our original imitated observed data from our previous paper using our psychiatric sample. In this case, we've expanded the sample to 333. And we find that if we do this similar approach now into that sample, we get what looks very much like a spectrum of activation fitting into the S-shaped pattern. And so moving forward, we hope to take this sort of advanced approach looking at this new way of looking at similarity space and applying it to our clinical populations to better understand brain system differences both in psychiatric and non-psychiatric patients, but also how that loads on people's social cognitive abilities, which is what this task taps into. That's the end of my primary slide. So for conclusions, activation patterns are highly variable. I know we all kind of know this, but it's worth repeating over and over again. fMRI task activation varies at least in part along this kind of negative to positive axis. This seems very prominent across tasks. This could be related to attentional effects. We consider the possibility it was hemodynamic, and we don't think that's the case. My personal preferred explanation is that this is kind of a behavioral neurostrategy effect. It's what people are doing in the scanner that drives this variability. And this bootstrap component space, it sort of represents more of a similar manifold space. It's a new way to look at variability across participants, and this space does relate to cognitive abilities outside the scanner. So that's all. I'd like to thank you all for your attention. If you have any questions, I'd be happy to answer them now or offline. And again, refer you to the preprint for further information as desired. Thank you very much.